Welcome, everybody. Today, it's a pleasure to have Neil King as my guest. Neil is an assistant professor in biochemistry. He's also a faculty member in the Institute for Protein Design. And perhaps more relevantly for this conversation, he is the scientific co-founder of Icosavax, which just went public a few months ago. Neil, welcome to the program.
Thank you. Happy to be here.
That's wonderful. So, we tried to find out a little bit about our innovators when we start this series. So tell us a little bit about yourself. How did a young Neil King become interested in biochemistry, in life sciences, or in computation?
Yeah. So it's a long story. So growing up as a young child, I come from a family of engineers, and my dad told my brother and sister and I probably 1,000 times before we turned 18 that when we went off to college, we could study whatever we wanted as long as it was engineering. And so I really didn't have much choice in the matter.
What type of engineer was your father?
So he was a civil engineer and built like large industrial processing plants and power plants and things like that. My brother, sister, and I ended up as chemical, mechanical, and biomedical engineers. And I just found myself drawn to biology. And then what happened was my senior year of undergrad, I took a course, Biomaterials 344 at Northwestern with Phil Messersmith, and he assigned the only research paper that I ever wrote in undergrad. Engineers don't write a lot of research papers. And I wrote mine on biomaterials for gene therapy. And this was just a few years after the death of Jesse Gelsinger from a viral vector gene therapy trial. And so there was a lot of backlash at that time against viral vectors. But the power of proteins in the space of delivery was already evident. At the same time, I was taking my first biochemistry course and learning about proteins, which are, of course, mind-blowingly amazing. And so these two things happening at the same time kind of turned this light bulb on in my head. And I said, okay, this is what I'm going to do with my career. I'm going to develop non-viral protein-based biologics delivery vectors. And that set me on the course that I've been on ever since.
Wow, this is great. And so, you actually came to the University of Washington. When was that? Was that in 2010?
2010.
2010, right? And it was before actually the Institute for Protein Design was created. And you had a big role in that because at the time, David was thinking a lot about protein folding, and less so about protein design. So you want to tell us a little bit about this as well?
Yeah. So when I joined the Baker Lab in the fall of 2010, it was right when Sorel Fleischmann and his colleagues had just designed the first de novo protein binder. So protein A that binds to protein B. And that, of course, was an incredibly exciting moment. No one had done it before. And if you can design de novo protein binding, of course, that unlocks this whole new area of protein design and engineering. And what I had been dreaming about since 2004, so for the last 6 years during my entire PhD, was designing new self-assembling proteins. And I wanted to do it through interface design, build these things the same way that Mother Nature does, which is through a combination of symmetry and non-covalent protein-protein interactions. So that design breakthrough that the Baker Lab had just made, I combined with Rosetta's existing ability to model symmetric arrangements of protein structures. And you combine symmetry, protein-protein interface design, and we came up with a general computational method for playing Legos with proteins, basically.
Right. And there was this 2012 science paper, I recall. I mean, this was a groundbreaking paper, and again, congratulations on that. After all these years, it's still very important. And so, you went up and you started thinking about building these more complex architectures, including icosahedrons. And Icosavax actually derives its name, from icosahedrons. Tell us a little bit more about this, and more importantly, when you were thinking about vaccine design and so on and so forth, there was at the time quite a bit of work on virus-like particles, and maybe you can explain in layman's term what VLPs are, and really how the concept that you brought forth was different.
As you say, there was a long history of people using VLPs, virus-like particles, as scaffolds in vaccine design. And the underlying idea here is that your immune system has evolved to react strongly against things that are repetitive. right? So viruses themselves are repetitive symmetric arrangements of proteins. Bacterial cell walls have repetitive chemical elements. So repetition is seen or perceived as a danger signal by the immune system. So, one idea is to present whatever antigen you want to target in a symmetric repetitive array, and maybe that will make a better vaccine. There's already clinical validation for this modality. The HPV and HBV vaccines are both self-assembling proteins. They are virus-like particles, which are capsid-like arrangements of viral proteins that lack the viral genome. So they're very safe. There's no risk of infection. There's no viral genome present, but they present their antigens in this dense symmetric repetitive array. And those are some of the best subunit vaccines we have, best vaccines made out of purified proteins that we have. They're very safe, very effective. However, what if you have an antigen like the SARS-CoV-2 spike that doesn't naturally self-assemble into a VLP? Well, then you need a heterologous scaffold upon which you can display that antigen. So people had been using naturally occurring VLPs, things like the hepatitis B surface antigen, bacteriophage virus-like particles, but these scaffolds were only capable of displaying short linear epitopes, tiny little fragments of an antigen, but you could never take the SARS-CoV-2 spike and display that on a bacteriophage VLP. And that was where our technology came in. So after that initial science paper in 2012, we increased the complexity of the materials that we could design. And a key aspect of that was in the initial paper, we were designing homomeric materials. So every subunit is the same in the large complex that you've made. What we then went to, and this was always the plan, do homomeric first, and then go to what we call two-component protein nanoparticles, constructed from two different building blocks or two different Lego pieces, if you want to think of it that way. The advantage here is you separate expression of the protein, production of each protein from assembly into the particle. And by separating those two processes, you can put very complex antigens like the SARS-CoV-2 spike onto these particles, and you can manufacture them at scale.
Absolutely. And that was, so it was fascinating. So, you came up with this idea and you started working on it and you increased the complexity and you made the particles that you wanted eventually. And this particle actually turned out to be very effective at inducing an immune response. And you tell me this was all part of the plan. That was part of the plan in 2008, 2005. I mean, this is pretty remarkable. Science in my lab has not necessarily worked that way.
So I have to clarify, and this is an important point. It was all part of the plan except for the whole vaccine bit. Because if you recall, I wrote that research paper in undergrad on biomaterials for gene therapy. And delivery was the goal that I had always been working towards. So I wanted to build these materials as vehicles to package and deliver medicines to the spot in the body where they need to go. And that had always been the motivation. And it was only once we had these nanoparticles, these materials, that we were having a conversation with Linda Stewart at the Gates Foundation. And she said, hey, these things would be great scaffolds for vaccines. And that was something I had never seriously thought about, right? And we started looking at it and we're like, well, they actually probably would be really good scaffolds for vaccines. And so then I faced a decision where I had to make the call between staying on the path and going after biologics delivery, which at the time I thought was going to be a long road, and still, 6 years later, 5 years later, I still think that's going to be a fairly long road, or pivot into this area of research that I had never considered, vaccines. And thinking about it, I was like, vaccines are some of the most successful medical interventions of all time. You're preventing disease. You're saving many, many lives a year. Am I willing to make that pivot? Yes, I am, right? And I just did a hard right turn into vaccines.
Yeah, and to put things in perspective, actually, you made that decision to go into vaccine, right, in the mid 2010. And a lot of companies were shying away from vaccine at this time, because people felt that there was no money into it. This was, of course, pre-COVID. And I remember that it was at about that time also that DARPA was investing seriously into nucleic acid vaccine, right? I mean, so, there was like a lot of different competing technology, I would say that were being developed. And so, how did you feel in this environment? You're going after, impact here. You're not going after money, and you have competitors that perhaps are going to bring forth a completely different approach to what you're trying to do.
Absolutely. No, it's a really good question, and it's an accurate depiction of the environment at the time. I think the reason that I was willing to make that right turn is I actually view vaccines as a delivery problem. You're delivering an antigen and maybe an immunomodulatory signal to a certain type of cell. You're delivering it to cells of the immune system, right? Now, this is very different than delivering CRISPR to, some very specific cell type. But nevertheless, they are both delivery problems. And so I thought, this vaccines are a twin application to delivery. And whatever we learn through the research that we do in vaccines, we will be able to come back in the end and apply this to the delivery problem. The other thing that I really liked about the vaccine space at the time was it completely circumvents the immunogenicity problem. So proteins are the most amazing molecules in the known universe. They can do anything, but the Achilles heel of using proteins as delivery vehicles is that you will get an immune response against your protein-based delivery vehicle. And that's going to be an issue that you're going to have to get around. And we're seeing this with the AAV, right, or the adeno vaccines for COVID and for all the AAVs that are being widely deployed in gene therapy. In vaccines, not an issue. And in fact, an immune response is exactly what you want. And so you're swimming with the current instead of against it. But again, anything we learn about how the immune system perceives these protein-based nanomaterials, we can then go back and apply in the delivery space. So even though there was competition, mRNA was gaining steam, but not yet fully established, we thought that there was a significant opportunity here. And we did a couple experiments, and the early experiments worked in preclinical animal models, and that gave us confidence to continue.
That is great. And so, one of the things that our innovators want to understand, if, you're a postdoc, if you're a senior graduate student, you've been working for a while, on a particular technology, or if you are a faculty member, what is the path? I mean, what is the decision, that made you want to create Icosavax?
Right. So yeah, that's a really important point. And it's something that at the Institute for Protein Design in the early days, we really debated a lot internally. At what point do you spin a technology out? Because you can do that too early and you can do it too late, right? And for Icosavax, I remember the moment well. We were on a call with the Gates Foundation in which they brought in an external advisor, and that was Barney Graham from the NIH's Vaccine Research Center. And the first indication in which we got preclinical data was RSV. And of course, Barney had spent his entire career working on RSV, and together with Peter Kwong and Jason McClellan, designed the pre-fusion RSV-F candidate, DSCAP1, that is now in clinical trials and advancing in clinical trials and looking good. So Barney really was an expert. And when he saw the data that we had in mice, the Gates Foundation asked him, Barney, do you think they should do an NHP study? And really what they were asking is, should we fund them to do an NHP study? And Barney's response was very interesting. He said, you can go do an NHP study, but these data in mice look so good and the competition is so fierce here. I think you need to get this vaccine into humans. You should go straight to humans. And in that moment, Icosavax was conceived.
Was there any surprises on the way, or is that always happening so beautifully?
I guess it wasn't too much of a surprise, but the thing that has impressed me the most is just how many people have to be involved in order to really take a technology over the finish line. And That is true both at UW and the Institute for Protein Design, but also at Icosavax. And the team that they've got at Icosavax pushing this forward is just incredible. It's an incredible team. And that, I think, is maybe the thing that has surprised me most is just how much effort and from such a large number of people is required.
Yeah, so talking about Icosavax, the company was founded, I think, in 2017. Is that correct?
Yep.
And you raised your first Series A in I've got these notes here, October 2019,
Yes.
and that was not a meager Series A. I think that you guys raised $51 million. That was huge, right? I mean, did you expect that?
And as you said earlier, that was a very large raise for a vaccine company at that time, because, as you alluded to, venture capitalists have never been too enthusiastic about vaccines, mainly because pre-COVID, the development timelines were so long. I mean, easily 7-10 more years to get to a product and start generating a return on their investment. So it's not a space that generated a tremendous amount of enthusiasm. So actually, this is another kind of hurdle and sidestep that we had to overcome. Icosavax got turned down by probably 40 plus VCs before we closed that Series A. It was a huge amount of work to put that together. And it took meeting the right type of investor who really believed in the power of the platform. Not a single asset, but the nanoparticle platform. And Mark McDade at Chiming was that person, saw that potential, and stood up to lead a Series A investment. And then we ended up rounding up a fantastic syndicate that put together a Series A that was going to give Icosavax the space and the resources to go be successful. But it took a lot of work, and the CEO of Icosavax, Adam Simpson, did an amazing job.
October 2019, right? Nothing is happening yet. Nobody has heard about COVID-19, but, some news are starting to permeate from China, and people are saying to worry. Certainly in January of 2020, people know something about this. And Icosavax is in a perfect space now because, people realize that they need a vaccine, specifically as March roll on. And, looking at the acceleration, that was fantastic, right? During this, year plus, you are basically, working on something that is absolutely necessary. How does it feel? You're thinking that you're actually working for saving the world, right?
I got into science and I pursued a scientific career because I wanted to contribute to inventing new types of medicines. That's what I wanted to do. I never wanted to do it in quite this way, right? But the situation came up. We had a technology that we think could be helpful, and we felt obligated to try to help. I had actually been working on coronavirus vaccines with David Wiesler's group here in the biochemistry department since 2017. So we had been putting the spikes from SARS and MERS on our nanoparticles. And so when SARS-CoV-2 broke out, we said, okay, look, we've got to jump on this, but we also need to try something a little bit different. And so we tried putting not only the spike ectodomain, but also just the receptor binding domain onto the nanoparticles, because we had gotten enough data by that time with other antigens to know that we could take a weakly immunogenic antigen, put it on our particles, and turn it into a very potent vaccine. And that's exactly what we did with the RBD. And so there was very much, as you mentioned, a sense of purpose. And that pulled the team together in a way that I have never observed before. The team here in the IPD core labs and in my group and in David Wiesler's group that was working on that vaccine in 2020 was the most remarkable team effort I've ever seen. There was no daylight between anybody. People were working extremely hard, not a single word of complaint. Everybody felt driven by that sense of purpose.
And things happen fast also on the fundraising level. In January of 2021, was it, or in April, I'm sorry. Now, I have my cheat sheet and I'm looking at it right now. You raised 100 million in Series B. Again, a very, very, very large raise. But more importantly, you were able to go an IPO in July, and it was $182 million that was raised there. So, this was a super accelerated timeline. How did it feel, for you as a founder to see the evolution of this company and things moving so fast, right?
Yeah. So I think the major, it's tremendously exciting to see Icosavax gain the resources to really develop a pipeline.
Mm-hmm.
Because again, we felt the whole time that we had a nanoparticle platform, not a single vaccine candidate, not a single asset, but a platform. And again, we were very fortunate that Icosavax's investors agreed, and they wanted to see the company reach the potential of the platform. And COVID turbocharged everything across the entire vaccine development space. And it turbocharged Icosavax as well. And I think the response to COVID generally, beyond Icosavax, highlighted the importance of technology platforms. The S2P mutations that stabilize the pre-fusion spike trimer were identified in 2017.
Mm-hmm.
And the existence of those mutations before the pandemic was an antigen design platform that all of the various vaccine makers were able to utilize immediately. mRNA, we've all heard the story. mRNA had been in development for 2 decades before SARS-CoV-2. And thank goodness, because that platform existed and was able to respond. Likewise, we had our designed protein nanoparticle platform. But seeing it kind of grow into its potential and seeing Icosavax execute on their plan and their vision to advance not a single asset, but really the platform, has been tremendously exciting. And it's because of the potential impact, right? That's what we're all going after, is we want to go make vaccines that save lives. And that Series B and that IPO, the proceeds from those fundraising events, is going to allow them to build that pipeline and advance it and go make vaccines that save lives. And it's tremendously exciting.
So you mentioned RSV as one of your next targets. What else are you working on?
So at Icosavax or at the Institute?
I understand that there are some things that you cannot tell me,
right?
But which are the targets that you feel are important? So, respiratory diseases, what clearly are going to be important. Do you think it might be equally effective, for some other type of infections?
Yes, I do. So Icosavax has a pipeline with several candidates for respiratory viruses, right? RSV, HMPV, influenza are all publicly disclosed targets that they're advancing. They do have additional targets that they're advancing that are not publicly disclosed. But here at the IPD, we've been applying this platform to a number of different indications, mainly in collaboration with the Gates Foundation, including global health targets like malaria, right, which is caused by a parasitic infection. We've done bacterial targets, we've done all sorts of targets, and we've convinced ourselves that this nanoparticle platform really is a robust and versatile platform. And again, it's because it's tapping into that evolved property of the immune system that sees repetition as a danger signal. So you're really plugging into something very fundamental in that host biology. And then the 2 component nanoparticles that we've designed, again, made manufacturing so much easier and more straightforward. And that's really what has enabled the clinical development.
Tell us a little bit, how did CoMotion help you along the way?
CoMotion was instrumental to the entire thing, right? So as you know, launching a company, attracting investment requires an IP portfolio, right? An intellectual property portfolio, because the investors want to know that there's something unique that this company has that nobody else has, and that's going to be this company's competitive edge. And so, from the beginning, because the idea was always to translate this nanoparticle platform, we filed patent applications through CoMotion at the University of Washington. And of course, I had never done that before. This was a totally new world, and it is a very different world than, protein design or protein structure. And so I feel like I have gotten an education I won't quite claim a PhD, but I've got an education in
JD.
Yeah. Oh, goodness, no. Goodness, no. But maybe a master's, right? In intellectual property from my colleagues at CoMotion. So in particular, Dennis Hansen has just been instrumental and has really taught me a huge amount about how do you think about IP? What is the purpose of IP? You have to have a reason to file a patent application, right? And in our case, the reason was we want to make sure we've got that portfolio to attract investment to a nascent startup, right? But Dennis helped me think through what are the reasons, when is it appropriate to file IP, and when is it just not a great idea? What types of IP should you file, and how do you go about doing that? The other person that was really instrumental was actually external counsel, Dave Harper, was amazing. In the early days, internal counsel at CoMotion, Andrew Laughlin, was absolutely incredible in terms of teaching me, how do you prepare? What is a patent? What does it look like? I had never looked through one. How do you write this thing? How do you translate science into legalese, right? To file this application. And then that's all kind of on the front end, putting together that IP portfolio. But then the other piece, of course, is actually licensing it, which is the process by which that IP leaves UW and goes out to these companies so that they can exercise the invention and make the thing and get it out for public health impact. And again, there, I think, Dennis, Fiona, others at CoMotion were really instrumental in terms of defining a licensing strategy that would enable Icosavax to be successful.
And we're blessed with a fantastic staff indeed. I wanted to ask you, what advice do you have for the budding entrepreneur who is watching that video right now? What should they do? What should they think about?
Yeah, I think, it's a great question, and everybody's path is going to be different. I think the things that I would say are pick a big problem, go work on a big problem, right? Find something important and relevant. And then the second thing I would say is embrace your own style. Even in science, people have different styles of doing science, right? And I think if you know your own strengths and weaknesses, you are able to play to your own strengths, bolster your own weaknesses, and then approach science with your own style. And that's how you're going to be maximally successful. So just embrace who you are, go for it, and then have a clear goal that you're working towards. It needs to be very, very clear. And if you want to start a company, there needs to be a purpose for the product that the company is going to make, and there needs to be a market.
Neil, I very much appreciate you taking the time to talk with us today. It was fantastic, and good luck to you.
Thank you so much.